All right, guys, this should finish up our cell communication notes packet. So if you were in class this past week, Tuesday or Wednesday, you know that we have talked about the cell communication pathway has three primary components: reception, transduction, and response. And we have talked our way and taken notes all the way through reception, and it's time to talk about transduction. And we did our POGL on the front end of our notes this time to really kind of guide us through an understanding of what is transduction. So if you're a little bit lost in this lecture, if you feel like, well, she's just plowing through information, it could be because you missed class or you are missing a bit of an understanding from the POGL. And if that's the case, I would encourage you to reach out to me. So find in your notes where it says B, transduction, and here we go with the transduction portion of the pathway. So the first thing that I want you to know is that the majority of the signaling pathway, specifically the transduction and even the response in a little bit, are going to be things that happen inside the cell. So these are intracellular processes that are occurring. So over here in our diagram, up here we have our ligand binding to the receptor outside of the cell in this case. Ligands don't always bind outside of a cell. Sometimes they can come into a cell and bind to an intracellular receptor. But in this model, ligand binds outside. And then all of this stuff happening right here, this almost like leapfrog looking event occurring with all of these proteins, that's happening intracellularly. And that collectively is a part of transduction. So the big goal in transduction is to amplify the original signal, which was given by the ligand. One little ligand binds, but we need to amplify that signal, essentially get on the speaker inside the cell and say, hey guys, it's time to do something, and ready an army of workers. In this case, that army of workers are proteins. And so the goal here in transduction is to amplify the signal and eventually establish this body of workers, active proteins that can go do something. And this process is accomplished really in two ways. So phosphorylation is one, and we saw that in our POGL. The process of phosphorylation, I'm going to make sure that you understand what that is. And the idea that one protein can phosphorylate another, that can phosphorylate another, we get sort of this multiplication effect of activating a bunch of working proteins at one time. And then also the use of what are called secondary messengers. So that's really how we are amplifying the signal. And so your notes moving forward, we're going to talk a little bit about that word that you just wrote down, phosphorylation cascade. And then we're going to also talk a little bit in the follow-up slide about the last bullet that you wrote down there about secondary messengers. So let's go with the phosphorylation cascade first. So if you're kind of confused, where am I writing this? It's still within this section of the notes, but you might want to sort of diagram a line off to the side of the word that you wrote down from this slide here, phosphorylation cascade, because I'm getting ready to explain what it is. Same thing in a couple of slides when we do secondary messengers. So let's write down a few things, and then I'm going to come back to this diagram, because really this diagram it's very, very simple, but it has so many bits and pieces that can help us understand this phosphorylation cascade. So first, what is it? Now, your POGL told you that phosphorylation is anytime you add a phosphate. So if you're hearing the word phosphate in phosphorylation, good job. Anytime you're adding a phosphate to an inactive protein in order to activate it, to turn it on. Okay, so anytime we can add a phosphate to an inactive protein in this transduction pathway, we are going to activate that protein. An activated protein will go do work. That work, we will find, is collectively referred to as the response in the cell communication pathway. These phosphates that are added don't come from nothing. So they're actually donated by ATP. right? So we recognize that term ATP, it's cellular energy. It is free floating in the cytoplasm. It's been created by the mitochondria of the cell. So ATP, which stands for adenosine triphosphate, will donate one of its phosphates, and that phosphate can be added to an inactive protein. So let's jump down here to this diagram at the bottom. This is very similar or very simple sort of pathway described. So something happens, we look at these a lot in chemistry, something happens over here, a reaction occurs, and then here is the result, right? So products and reactants, if that is helpful to you as language as well. So for the kind of front side of this arrow, you see a substrate and you see ATP. So for that substrate, you could also substitute the word inactive protein. So you have an inactive protein and you have ATP sort of free floating in the cell. Now check out ATP. There's a red component, but then there are also 1, 2, 3 of these little phosphates. That's why ATP is called adenosine tri-3-phosphates. So these are free floating. Well, something occurs, and we'll come back to that something that helps it occur right above this arrow. Something occurs that allows an ATP molecule to chop off or cleave one of its phosphates, and that phosphate can then be added to the substrate or the inactive protein. So let's look at what happens once that has occurred. So follow the arrow over here. You have ATP, which lost a phosphate, now called ADP, adenosine diphosphate. Di is another term, di and bi can indicate two. So you have ADP, adenosine diphosphate, that now has 2 phosphates. Remember, it lost one that would have been right here in the format of ATP. And then you see that phosphate has been attached over here to this protein. And so now you have an activated protein. And it is all thanks to the work of this molecule sitting above the arrow. So kinase enzymes are a category of enzymes that will add a phosphate that has been donated by ATP in this case to an inactive protein. In doing that, it turns that protein on. Now, we cannot leave activated protein, this pathway on forever, because as you can see, it uses energy. It sort of breaks down ATP a little bit. And so as long as this diagram is running inside of a cell, as long as kinase is taking phosphates that have been removed from ATP and putting them on proteins, we are losing energy. And so to kind of shut down this pathway, we have another category of enzymes step into play. So this word is not reflected on the diagram. Phosphatase enzymes are a category of enzymes that will actually come back and sort of undo what kinase did. They remove this phosphate, and in removing the phosphate, this protein becomes deactivated or inactive again. And then this free-floating phosphate can attach itself back to an ADP that is floating around and can be converted back to ATP. So phosphatases are important in sort of shutting this phosphorylation cascade down and re-energizing ATP. All right, let's look at secondary messengers. Well, I should back up. So this entire process that we just talked about was all about activating proteins. You have to add a phosphate to activate them. Okay, let's do that. Anytime we are activating proteins, we are creating what I'm calling worker proteins, proteins that can go do a job. And that job might be directly connected to the ultimate response of the cell communication pathway, or that activated protein might go on to activate another protein to sort of continue the chain of events that are occurring in this phosphorylation cascade. But either way you slice it, the phosphorylation cascade is there to basically turn on, activate a whole bunch of working proteins so they can go do something. Now, secondary messengers are going to be helpful in this activation process as well. So secondary messengers are small, water-soluble molecules that can move around easily. Some of them exist outside of the cell and they have to make their way in. We saw that in the POGL with the calcium that was extracellular that had to come through the protein receptor to get into the cell. Some secondary messengers already exist within the cell, stored in specific organelles. They just need to be released, or they can be created using other molecules from within the cell. So regardless, what I want you to kind of latch onto here is that secondary messengers are small and they can move around pretty easily. They can come into a cell, they can exit a cell, they can come into and out of organelles pretty easily. And here's what they do. They are going to help. They act as a support system to those kinase enzymes that we talked about just a second ago. So they will help kinases continue to activate these, what I'm calling worker proteins. Okay, so large net effect here in the transduction pathway is that you have a phosphorylation process happening by protein kinases. And then you also have these secondary messengers coming alongside and supporting the kinases to further activate additional proteins. All that means with both of these working at the same time is you get more proteins activated in a shorter amount of time. There are two really common examples that are often talked about in these models as secondary messengers. And so I wanted to put it out there to you. Calcium is a very common secondary messenger. In this diagram down here, you can see calcium that is being released, actually in this case, from the endoplasmic reticulum, that's going to go bind on this protein called the PKC and activate it, turn it on. Another really common secondary messenger is something that looks like the word camp, cyclic AMP, and that's up here in this diagram. So cyclic AMP can be created, once again, from cellular ATP. And once it is created, it can go on to activate proteins to enhance the cell's response. So if we kind of move ourselves to the end of this and talk about the response, so back to this diagram here for a second, remember the three basic steps in any cell communication pathway: reception, transduction, and response. It's time to talk briefly for a moment about response. The first and the last part of a cell communication pathway are always pretty easy to identify in a model. What starts the process and what is the end result. Everything in between then becomes transduction. So let's focus on that response. It is as it sounds. This is the cell's final response to the overall communication pathway. So when you're trying to figure out what that response might be, here's a question that I would encourage you to ask yourself. What did all of those activated proteins, the things that I just spent time activating with kinases, what did those activated proteins actually go do? And so we often find these responses lumped into two broad categories. There are responses that exist inside the nucleus. These often relate to expressing DNA or not. So these are going to be like, do I turn a gene on? Do I turn a gene off? Which really means, am I going to create a bit of protein based off of a region of DNA, or am I not going to do that? So those are nuclear responses. They typically involve expression of our genome. And everything else is lumped into cytoplasmic responses. So that's anything that's happening out in the cytoplasm, in the cytosol even, things like building up and breaking down these molecules, macromolecules even. And so thinking about responses, and really multiple responses could occur as the result of one ligand binding to a receptor. So you might have to kind of look in some of the models. We'll practice this the next class time that we're together this week to see what are all the possible different responses that might exist. Some inside the nucleus dealing with gene expression, and then some outside of the nucleus in the cell dealing with other aspects, metabolism, cellular life and death of the cell. Now, I want to take a second and pull all of this together by looking at a specific example. So I want to talk about our fight or flight response. And so fight or flight, you've probably heard when you see something that is maybe startling, makes you scared, makes you angry, your body responds. And typically, it's actually now fight, flight, or freeze. We've added freeze to it. But typically, you're either going to stand your ground or you're going to run away. And when we think about the ligand that is triggering the majority of the things that happen in a fight or flight response, it is epinephrine. Epinephrine is like a buddy to adrenaline, if you've ever heard of adrenaline. And you might have even heard of epinephrine relating to like an EpiPen. If somebody goes into, has a severe allergic reaction, they might have an EpiPen that they kind of shoot into their thigh to cause some of these effects, particularly in the lungs, right? That we're going to see in this video. So in the video, I want you to focus, the ligand is epinephrine, and I'll help and make sure that you see the ligand. Now, what's really cool about this video is that this ligand does a lot of different things in the fight or flight response. So you think about when you are scared or anxious, afraid, lots of things happen in your body. You might start to sweat. Your mouth might go dry. The hairs on the back of your neck and your arms might stand up. Your pupils dilate. So there are lots of different things that epinephrine actually, your breathing, your respiration increases, your heart rate increases. That is all because of what epinephrine does when it lands on receptors in specific organs. And so for us, you're going to see several of those responses here in the video, but for us, we're going to zoom in on the target organ and the target cell what happens when epinephrine lands on the liver. So your liver, as a reminder, is where you store a bunch of glycogen. Glycogen is that really big complex carbohydrate and is really stored for later as an energy source. So the target cell here is going to be the storage center of glycogen, the liver. And so we look at what happens when epinephrine lands on the liver and the process of taking the glycogen that is stored there and breaking it down and releasing it into the smaller unit of glucose that is an energy source for fight or flight. So either way, if you're going to fight, you need energy. If you are going to flight, you need energy. So either way, we're talking about an energy source that is required. Okay, so I am going to make sure that my audio doesn't cut off as I start to play this.